So we're this is part 3 of lecture 2 of bio 310. Um we're talking about DNA repair mismatch errors now. Going to that, I believe I left off at the clamp loader. No, I was going over the entire bacterial replication sequence. Yeah, entire bacterial replication sequence, and it started with the helicase opening the DNA, SSB protein stabilizing the strands, primase makes the primer, sliding clamp attaches, DNA polymerase copies the DNA, and like leading strand continuous, lagging strand Okazaki fragments, ligase joins the fragments, and then mm, we went over all that and we spoke about how the difference. We spoke about the difference between a bacterial replication fork and a eukaryotic replication fork. And then we came to DNA repair and mismatch errors. Sometimes DNA polymerase misses a mistake instead of ET. Oh, we have been over this as well. And then we did double strand breaks, right? This is much more dangerous. Instead of one damaged base, there's both DNA strands broken. Well, for before here, right? If there's a mismatch error, all it simply would do is like switch this out. So DNA polymerase would stop. It would take it out and put the right one back in, and then ligase would glue it back together. Right, that was simple. But with double strand breaks, it's not as simple. Um, there's two repair methods: either non-homologous end joining, NHEJ. That reminds me of my forever, but that's how I'm gonna remember this, right? NHEJ, which is non-homologous end joining. It's fast, but it's not accurate. So for that one, step one would be to just processing of the DNA end by nuclease and joining by DNA. So it's basically just cutting them, right? And then it's putting them back together, deletion of DNA sequence. So you're deleting a lot of stuff here, right? So nucleotides are lost, and the ligase joins it back together. So it's done. But this is not the most accurate, which is why we go on to the second repair process, repair option, which is homologous recombination. Homologous recombination is slow, but it's very accurate. So here, as the picture goes, you have damaged DNA molecule, and then you have undamaged DNA molecule. All it does is processing by broken ends by recombination specific nuclease. So you're kind of breaking it here, right? But it's recombination specific. So a double-stranded break accurately repaired undamaged DNA. So this is they kind of, even though it's broken, they can fix it in a way that it's back together as it was before. So it's very accurate. Cell copies and copies information from the undamaged sister chromatid. So you see here, this is undamaged. It's just copying that copycat killer. Like you know how Dexter in Dexter there's a copycat serial killer. I have been watching way too much of Dexter. Anyways, um, but here it's like it copies this undamaged DNA and it uses it like a blueprint. So broken DNA becomes exactly the same as before, no mutation. Think copy homework from your twin. So that I guess it works too. So compare NHEJ. NHEJ is non-homologous end joining, right, Lily? Yeah. It's fast, error prone, no template, whereas homologous end joining, it's slow, but it's very accurate. But it needs another sister chromatid, which is undamaged. Now we're moving on to misDNA repair leads to mutations. DNA replication can make mistakes, mismatch repair fixes it, original DNA restored, genome stays stable. If repair fails, the wrong base remains. So repair fails, the wrong base has remained. AG is not supposed to be together. It should be AT, right? So wrong base remains. Next replication copies the mistake, right? It's copying the mistake. Now mutation has become permanent. Possible consequences: no effect or genetic disease, which can lead to cancer and evolution. Mutations are not always harmful. Some are natural, some are useful over evolution. Now, okay, if we look at the diagram here, you have the parent DNA molecule, DNA replication happens, new strand with error, you have an error, right? Through mismatch repair, we try to fix it. Original DNA sequence is restored. You're back to the original copy. You're giving the original copy, and then you have a mutated copy, 80, which is not necessarily wrong, but it is the mutated copy, and it's a different combo, right? Possibly no phenotype will result. It can also result in, like, it's not wrong. AT is not wrong, but it's not the same as parental. So that's why we're saying there is a chance that there might nothing might happen, but there's also chances of genetic disorders, cancers, and individual variation and evolution. Now we're gonna move on to CRISPR-Cas9. It's one of my favorite topics. It's basically when I remember this by the Chinese guy who tried to fix HIV. I think he did kind of fix it, HIV, but he basically um he he evolved not evolved he before the mother even gave birth, he altered the embryos, the child's DNA in such a way that HIV was prevented, and the kid was born with HIV, no HIV, even though the mom had it, right? So that's one of the unique ways CRISPR-Cas9 was used. But one of the concerns is that you're like ethical concerns, like you bring God, etc. But back to what it's actually used for, CRISPR-Cas9 technology takes advantage of cell's ability to repair double-strand breaks. So we're gonna move on to how it actually CRISPR-Cas9 actually works, right? Um, it's a more this is modern gene editing. Scientists copied it from bacteria. Cas9 is a protein. It acts like molecular scissors. It cuts DNA. Cas9's job is to cut DNA. gRNA is guide RNA. It works like a GPS. It finds the exact DNA sequence. Cas9 follows it. PAM sequence, Cas9 cannot cut anywhere. It first looks for PAM, usually NGG. No PAM, no cut. Right, so we're looking right here, right? You have Cas9 as a protein, gRNA, you have a little cleavage, right? The target is here. If there's no PAM, there will be no cut. Here there is PAM, so there was cut, and then you have the cut. Step one is to guide the RNA to bind to target RNA. So guide the RNA to bind to target RNA. Cas9 recognizes PAM. Step three, Cas9 cuts both the DNA strands, not just this one or just this one, both of them. And then the cell now repairs it. So there's two choices. These two, right? Repair one, non homologous end joining. Oh, do you see this? It's just cut and then they're glued back together. This is non homologic, this is end joining. And then on the other hand, you have homologous end joining where you're looking at a sister chromatid and you're basically copying it, like copying a homework copy for to win, and then you get like this. This is slow but accurate. This is fast but inaccurate. So let's go over NHEJ quickly. The cell quickly glues DNA back together, usually introduces insertions or deletions. This often knocks out the gene. The scientists use this to turn genes off. Repair 2, homology directed repair, HDR. Scientists also provide donor DNA. The cell copies from the donor DNA results precise gene editing. Scientists can fix mutations, insert genes, replace genes. So I have a little something I'd like to share. If I can find the right tab. Here we go. Final comparison, high yield. We speak about all of their functions. Seeds, SSB is holding the strands apart. Sliding cap holds the polymerase on the DNA, so they're different. NEHG, gRNA guides Cas9 to the PAM recognize required for recognition before Cas9 can cut. So that's PAM is pretty important. gRNA guides Cas9 to target DNA. So like Right here, you're seeing gRNA guiding the Cas9 to target DNA, so Cas9 can do the cutting once it recognizes PAM. So you first order of business, gRNA, PAM, Cas9, cut, two different outcomes. So this is something that we've already kind of went over, but it's for you to read at your own leisure. That'll be all for lecture 2. Thank you for your time. Oh, also, PAM is proto spacer adjacent motif, NGJ, whatever that means.